So, good day everyone. Today I will be talking about the Lutheran system, or known as 005 system blood group. So, the Lutheran system history. It was founded in 1945 in the serum of a patient with lupus erythematosus following the transfusion of unit of blood carrying the corresponding low prevalence antigen. So the patient also made anti-C, anti-N, and the first example of anti-CW. Lutheran comes from the donor's name Lutheran. So I don't know if it is a hoax or true, but they said that the donor's name was actually named Lutheran, but had been misspelled as Lutheran on the sample tube. During 1965, Bush and Chanarin described anti-LU, which defined as the antithetical partner to LU. In the year 1961, when Crawford and colleagues described the first LU A negative and B negative phenotype, so the blood group system appeared completely until 1961. Unlike most null phenotypes at the same time, this one demonstrated the dominant inheritance. So in the 1963, the LU A negative and B negative phenotype inherited as a recessive silent allele was described. So there are 20 antigens are part of the Lutheran system. They are numbered through LU22. So the LU10 and the LU15 was obsolete. Most of these antigens are high prevalence, but four sets of antigens are antithetical. Antithetical means that a pair of antigens that are coded by different alleles of a single gene. So So the antigens are high prevalent, so only few people lack of antigens and can make alloantibody, or very low prevalence, so that only a few people are ever exposed. Consequently, the antibodies are seen infrequently, and there are not much data on the clinical significance of Lutheran antibodies. So the molecular basis for the four pairs of antithetical antigens and several of the high prevalence antigens has been determined through the creation of the Lutheran glycoprotein mutants and subsequent sequencing of the exons encoding the extracellular domains. For most of the antigens, expression was caused by a single nucleotide polymorphism resulting in a single amino acid changes on the protein level. So many of the antigens were associated with the Lutheran systems when their corresponding antibodies were non-reactive with the rare LU, A negative, and B negative RBCs. So the ISBT designed for the Lutheran blood group system is LU, or known as 005 system. So these are the Lutheran system antigens, or these are the summarize of those 20 antigens. LUA and LUB are antigens produced by allelic codominant genes. There are two major antigens, which are the LUA, or the low frequency, and LUB, which is the high frequency. There are also three further pairs of allelic codominant genes that control the LU6 and LU9 antigens, while the LU8 and LU14 antigens and the AUA and the AUB antigens 10 other high frequency antigens are pair of the system. So as you can see, these are the prevalence of common Lutheran phenotypes. So the number of LUB sites per RBCs is low. Next is the antibody characteristics. The antibodies may be stimulated by pregnancy or transfusion. The first antibody was anti-LUA. So IgM, naturally occurring silent agglutins that reacts better at room temperature than at 37 degrees Celsius. Among the relatively uncommon antibodies against Lutheran antigens, the anti-LUA is the most common. Most examples of anti-LUA or a few react at 37 degrees by indirect antiglobulin test. Some are capable of binding complement, but in vitro hemolysis has not been reported. Anti-LUA often goes undetected in routine testing because most reagent RBCs are LUA negative. The anti-LUA is more likely encountered as an incompatible cross-match or during the antibody workup for another specificity. As such, most anti-LUA will directly agglutinate RBCs at room temperature as well as in the 37 degrees Celsius I80 phase. Lutheran antibodies recognized by their characteristic loose, mixed field reactivity in the test tube. Examples of anti-LUA that react only at temperature below 37 degrees are clinically insignificant. There are no documented cases of immediate HERs. There are only rare and mild delay HERs due to the anti-LUA. Next is the anti-LUB. IgG and reactive at 37 degrees at the antiglobulin phase. Most examples are the antibody is made in response to pregnancy or transfusion. Allo anti-LUB reacts with all cells that are tested except the O2 control, and the reactions are often weaker with LUA positive, B positive RBCs and cord RBCs. Anti-LUB has been implicated with shortened survival of transfused cell and post-transfusion jaundice, but severe or acute hemolysis has not been reported. So the genetics of LU gene is located in chromosomes 19 at position of 9q13.2, along with genes that govern expressions of several blood group antigens such as H, SE, Lewis, LW, OCA, or the number of antigens are 19. So now let's talk about the LU A negative, B negative phenotypes. There are three genetics explanation or the mode of inheritance. First is the dominant type, the recessive type, and the recessive X-linked inhibitor type. First is the dominant type LU A negative and B negative. So the gene responsible for it is the erythroid propyl-like factor. The first LUA negative B negative family was reported by the propositus herself because the phenotype was seen in the successive generation in 50% of her family members and the others. And because non-individuals passed normal Lutheran genes, so their offspring, the expression of Lutheran was thought to be suppressed by a rare dominant regulator gene later called INLU or inhibitor of Lutheran. So a transcription factor were shown to be associated with the INLU phenotype in 21 of 22, and in LU individuals was studied. 49 in all cases, the mutated AKL allele or the gene responsible occurred in the presence of the normal AKL allele. This study concluded that the inhibitor Lutheran phenotype is caused by the inheritance of loss of function mutation on one allele of AKL, and they do not make anti-LU3. So we will be talking later on about anti-LU3. Other RBC's antigens affected are the reduced P1, INB, MER, and CD44. In addition, the reduced expression of the Lutheran antigens, dominant type LU A negative and B negative RBCs also can have reduced expression of CD4 and a weak expression of P1, I, ANWJ, MER2, and INB blood group antigen. Next is the recessive type LUA negative and B negative. So the gene responsible for it is the LU. In some families, the LUA negative B negative phenotype demonstrate recessive inheritance, the result of having two rare silent alleles LU-LU at the Lutheran locus. The parents and offspring of this null may be LU negative, A negative, B negative, but dosage studies and titer shows them to carry a single dose of LUB. They don't have LU antigens. It's either they have the null phenotype and can make inseparable anti-LUAB cold or cold anti-LU3. So, in other terms, they make anti-LU3. They also have a normal antigen expression of P1 and the other antigens that are weak with the dominant type. Last is the recessive X-linked inhibitor type. So the gene responsible of it is the XS2, the rare inhibitor that suppresses an hemigygous state. There have been no families reported with this rare X-linked LUAB phenotype. So an LUA negative and B negative phenotype in a large Australian family did not fit either the dominant or recessive inheritance pattern, but they are all male and carried trace amounts of LUB detected by the absorption or the elution, but still there are no other RBCs seen affected. Now let's talk about the anti-LU3. It is a rare antibody that reacts with all RBCs except LU A negative and B negative RBCs. The antibody looks like inseparable the anti-LUAB and recognized as a common antigen, LU3, that is present whenever LUA or LUB is present. So it is made only by individuals with the recessive type of LU A negative and B negative. Is the clinical significance. The transfusion. Both antibodies have been reported to cause mild or delayed transfusion reaction. IAT cross-match compatibility, blood should be transfused. The provision of LUB negative blood may be difficult, but the antibody is seldom present. For the hemolytic disease of the fetus and newborn, Lutheran antibodies have not been reported to cause severe HDFN or the hemolytic disease of the fetus and newborn, as the antigens are only weakly expressed on cord cells. So that's the end of my report. I hope you get something from this blood group system.